Hi everyone, so I just wanted to go over the phases of meiosis one more time. So I'm going to show you what the chromosomes are doing, what the cell is doing, and go through each phase. So to start, I have interphase. So this is before the cell is ready to divide. Interphase is part of the cell cycle. It is where cells prepare for division. And in this case, we have a germ cell. Germ cells are the cells that go through meiosis. They are the ones that start meiosis. They are found in sex organs, so ovaries in females and the testes in males. So you'll see that in the nucleus of this cell, each chromosome is not duplicated yet. It's still a singular chromosome. So during interphase, during S phase of interphase, each chromosome will get its sister chromatid, okay? So each chromosome goes through DNA replication. That whole process we learned about at the beginning of remote learning. DNA replication. Oh, that one's a little short. Let me find a better match. You might remember DNA polymerase and helicase and all those enzymes that regulate DNA replication. So now each chromosome has its sister chromatid, and this cell is ready to start meiosis. Okay? So now prophase 1. In prophase 1 of meiosis, lots of things happen. This is the important phase. First of all, the spindle fibers start to form. These centrioles should have been here already. That's one thing I forgot to draw. And the spindle fibers start to form. The nuclear membrane starts to break down. And the chromosomes condense. In this case, it's pipe cleaners. Can't really show them condensing all that much. What I can do is I will twist them to make them look like the X shapes you're used to. So this is just the sister chromatids that were made during interphase join together at the centromere. Remember, the centromere is the joining of the two sister chromatids, and they condense. Okay, so we have our chromosomes. Now that's what that's what we learned in mitosis, right? Mitosis, the spindle fibers form, nuclear membrane breaks down, chromosomes condense. There are other things that happen during meiosis in prophase that are really, really important. The first thing that happens is the homologous chromosomes synapse, or they bind to each other. So in this case, the homologous chromosomes are going to be sorted by color. So our purple chromosomes are going to be homologous. We can pretend that this is like chromosome number 1, right? So the purple chromosomes will synapse, meaning they actually join together and form that tetrad, right? They actually link up. It's like they're holding hands. We'll join the green and the blue. Okay, so these are our homologous chromosomes. So we know that the germ cell is diploid because each chromosome has its pair, right? There are 2 purple chromosomes, 2 blue chromosomes, and 2 green chromosomes. So this cell inherited one of each chromosome from its parents. So this germ cell would be located in a person, and that person got one dark blue copy and one light blue copy. Maybe the dark blue came from this person's mom, and the light blue came from this person's dad, right? All the chromosomes you have are from your parents. They're copies, they're actually from your parents' sperm and egg cells. So that's what makes this cell diploid. So during prophase 1, the homologous chromosomes synapse to form a tetrad. And the last thing they do is that process called crossing over. So these sister chromatids, or they're not really sister chromatids, these non-sister chromatids of the homologous pair actually swap genetic material. So a little piece from each chromosome actually gets swapped. So now a little piece of this chromosome is going to be on this one. I'm not going to do that. Do it this way. Okay, so now the light blue chromosome has a little bit of dark blue in it. And the dark blue chromosome is going to have a little bit of light blue. Okay? And that actually happens for all of the homologous pairs. I'm only going to show it for one just to save time, but that is what happens. The non-sister chromatids, so this chromatid and then this chromatid, wherever they link up, they're going to swap genetic material. And this process is random. It doesn't happen the same way twice. So there's no guarantee that like this gene and this gene, right, this gene on this chromosome and this gene on this chromosome are always going to swap. That's not the way it works. It is random, which is one of the ways that all sperm and egg cells wind up being genetically unique, okay? So now I'm gonna, by the end of prophase 1, your nuclear membrane is completely gone, and your spindle fibers will have attached to the homologous pairs. Okay? Oops. So now we finally get into the next phase, which is going to be metaphase 1. Okay, so I'm just going to redraw all this. Just to clean it up a little bit. So at metaphase 1, our homologous pairs will still be synapsed, and they will line up with their partner. It's like square dancing. They're lining up, they're do-si-doing. And they're holding hands. They're in that tetrad form still. Okay, so they're still linked to their partner. And remember that these would also have gone through crossing over as well. I only just showed it in this one. Okay, and you'll still have your centrioles with spindle fibers attached But this time, instead of attaching to the kinetochore and splitting apart the sister chromatids, in anaphase 1, it is the homologous chromosomes that get split up. So this chromosome and this chromosome get pulled apart. This one and this one, this one and this one. So it's the homologous pairs that get separated during anaphase 1, okay? And then you get that classic telophase shape. I forget which ones were on which side. Oh well. Whichever side they were on. Oh, I totally forgot to mention that. I'll go back and talk about that. It is important to have realized which side each one was on, because that's independent assortment, right? Whether this was on this side or this one was on this side completely changes the fate of the sperm and egg cells made at the end, right? So it is actually really important which one was on which side, but just because I forgot, I'm gonna leave it like this. I'll go back and talk about independent assortment again at the end. But in telophase 1, you still have your centrioles, but this is kind of tricky because depending on what source you look at, you'll see different things. So between telophase 1 and prophase 2, it would be wasteful for the cell to reform the nuclear membrane, break down the spindle fibers, and uncondense the chromosomes, when all of that's going to happen again in prophase 1. There's no interphase in between telophase 1 and prophase 2. So basically, if in telophase 1 the cell reforms, it would then just get completely broken down again in prophase 2. So it is kind of wasteful. So for our purposes, we're going to say that the nuclear membrane sort of reforms, the spindle fibers sort of break down, right? And the chromosomes sort of uncondense, right? They're not going to form all the way back just to be broken down again. So in this, it's kind of a gray area, okay? If you were ever asked a question about telophase 1, it would be very specific, because like I said, depending on what source you look at, you'll hear different things, okay? But that's telophase 1. And then of course you get cytokinesis, which splits these cells. And these cells are now haploid. They have only one blue chromosome, one purple chromosome, and one green chromosome when they started with homologous pairs. There's no homologous pair in this chromosome. This is a duplicated chromosome. It's not a homologous pair. This was a homologous pair, right? The light blue and the dark blue. This one is just it has a little piece of the dark blue, but this is just one chromosome, one chromosome, one chromosome. So this cell is now haploid, okay? So after telophase 1 and after cytokinesis, you get haploid cells, all right? So now, I'm gonna redraw this again just to make it look a little bit neater. We can say that the nuclear membrane is kind of back. Didn't really reform all the way in telophase 1. Now, meiosis 2 is essentially the same thing as mitosis, all right? So there is no interphase between meiosis 1 and meiosis 2. The two new cells made at the end of meiosis 1 go straight into meiosis 2. So the first thing is going to be prophase 2, okay? In prophase 2, the nuclear membrane goes away, if it ever came back at all. It really just depends on the type of cell. Okay, it's kind of a, like I said, a gray area. Spindle fibers reform if they ever went away. And chromosomes condense if they ever uncondensed, okay? Nothing else special happens during prophase 2. All the magic happened in prophase 1, okay? These chromosomes will get pulled to the center of the cell, right? That's kind of like late prophase 2. These spindle fibers have attached to the kinetochores and they'll yank the chromosomes to the center of the cell. Okay. And now we're in metaphase 2. Chromosomes are at the center of the cell. Spindle fibers are attached to the kinetochores, and now those sister chromatids get separated. I forget what side that was on. Let's put it on that side. Okay, so this is anaphase 2. Now those sister chromatids get separated, just like in mitosis. Okay? So those spindle fibers are pulling the sister chromatids away from each other to the opposite ends of the cell. And then, again, I'll redraw everything. So we're now going to have 2 peanuts. Now the nuclear membrane definitely reforms. The spindle fibers definitely break down. And the chromosomes uncondense. Okay, so that would be telophase 2. Right, you've got your nuclear membrane coming back, your spindle fibers go away, and the chromosomes uncondense. We don't have 4 separate cells yet, but you can see how 4 new cells are about to get made. You're going to have this cell, this cell, this cell, this cell. This one is going to be haploid. It's going to have only 1 blue, 1 purple, 1 green. And if you rewind to the beginning of this video, you'll see the original germ cell had 2 blue, 1 light, 1 dark, 2 purple, 1 light, 1 dark, 2 green, 1 light, 1 dark, right? But we've separated all of those chromosomes into these 4 separate cells, right? Here's the light, here's the dark. And then at the very end, another room. Okay, you get your 4 haploid gametes. Let's say these are sperm cells. There you go. That's meiosis. You wind up with 4 genetically unique haploid gametes, right? Remember that each of these would have a little bit of recombinant DNA, and it would have the effects of crossing over. They would all kind of look like this one. And that creates genetic diversity because it's random which genes get swapped, and you can see the result of independent assortment. What if during metaphase 1, all of the maternal chromosomes lined up on the left and all of the maternal chromosomes lined up on the right? The end result, you'd have all the light chromosomes together and all the dark chromosomes together. But the way they happened to line up, it was completely random. You have a dark and 2 light, a little bit of light and dark, light, light, dark, dark, light, a little bit of both, dark, dark. None of these 4 cells are the same. And in fact, no 2 sperm cells made in a male's life will ever be the same because of independent assortment and crossing over. Okay? Now let's say let's talk about conception, right? How does this then become a zygote or an embryo one day? So let's say that this sperm happens to find its way to an egg. I don't think I need to teach you how that happens. You can learn about that in a health class. So this sperm has come in contact with an egg. Eggs are much larger than sperm, and that egg is also going to be haploid because it was made by meiosis, and it has its own set of purple, green, and blue. Right? So it still has the same chromosomes, right? Every human has the same genes at the same location on its chromosomes. It has 23 different distinct chromosomes, and it has 2 copies of each. None of those 2 copies will be identical, right? And if you pick up, I used this blue for the male, this blue for the male during the whole meiosis scheme. So this egg has a completely different version of the chromosome than any of the male that made this sperm had. So this is a whole different set of chromosomes, but it's still the purple, the green, the blue. It's just got different alleles, right? Maybe this chromosome is carrying the allele for freckles, right? And this one is not. Get all that different kind of variation in all the alleles. But either way, this blue chromosome carries the gene for freckles. This one gives you freckles. This version of that allele will not give you freckles, okay? And it's completely random which sperm gets released from the male, because if you remember, they make 1,000 sperm every second. So this sperm just happened to get made, and then it was the one that was released, and it was the one that came in contact with this egg. So that's another way that offspring become kind of varied. This is why you don't look exactly like your parents, because this sperm cell, it's a one in an infinite, I mean, the number is infinite almost, you know, that this is the sperm that happened to come in contact with this egg. If males make 1,000 sperm every second, think about how many they make in a lifetime. And how many kids will a male, like the Duggars, what are they at, like 19 and counting? That dad has 19 kids, but you know, how many sperm did he make over his entire lifetime? And it's just those 19 sperm that happened to come out and met with an egg. So that, again, the selection of which gametes come in contact with each other adds, again, to genetic diversity. So, anyway, this sperm will come in contact with this egg, and it actually fuses. This is fertilization. This sperm releases its contents, its genetic contents, into the egg. And for a moment, this zygote has 2 nuclei, okay? If you actually watch videos of fertilization, if you watch like in vitro fertilization, they basically just take genetic material in a needle and inject it into an egg, and it looks like it has 2 nuclei at first, but it will fuse. And now it's diploid again. It's got 2 blue, 2 green, 2 purple. It's diploid. And then this zygote will start to go through mitosis to make the cells of the embryo, which will then continue to divide to make the fetus, and then at birth you have a baby. So this cell then starts to go through mitosis to make all the cells of the body. All right? So that is conception, that is sexual reproduction. Hopefully this communicates just how important meiosis is. If humans didn't do meiosis, then you wouldn't have sperm and egg cells to create future offspring, right? Because you can't have diploid cells come together, because then you'd have if you had 2 diploid cells fuse at conception, you'd have a mess. That's what you'd have. This is what you'd get with 2 diploid cells coming together. This is tetraploid, not haploid, not diploid, not triploid, but tetraploid. You have 4 copies of each chromosome. And when we talked about meiosis, when the first set of notes you did for meiosis, I talked about Down syndrome. Down syndrome is the result of having 3 copies of chromosome number 21. So it would just be like this. So if the cell was otherwise normal, but just had an extra copy of the blue chromosome, that would lead to a genetic disorder. There's actually lots of different trisomies, they're called. Klinefelter syndrome, Turner syndrome, all sorts of syndromes result from extra copies of chromosomes. So it is imperative that a haploid cell and a haploid cell come together, fuse, and then create this diploid cell, right? Just throwing in one mistake leads to very serious medical issues. So now I just want to rewind and talk about independent assortment one more time, because it is a concept that I think it's the hardest to conceptualize. So I keep talking about pairs of shoes, pairs of socks. Well, just so happens that I have the perfect demonstration for this topic. Okay. So imagine this is a cell. Okay. This cell would be diploid. You would actually start like this, right? This would be interphase. This would be interphase. Each chromosome gets duplicated. Each gets its sister chromatid. All right, then prophase, the homologous pairs synapse. The homologous pairs, we're going to use color to show homology. The pink chromosomes, the purple chromosomes, the blue chromosomes, right? So this right here, it's perfect that these have stripes, because you can picture this as a gene right here. On chromosome number 2, the purple chromosome, there's a gene right here. This chromosome at that gene has the allele for a widow's peak. On this chromosome, at that same location, the same gene, this one has the allele for not having a widow's peak, and that is actually a genetic thing. So perhaps this individual, their father gave them a widow's peak, their mother did not give them a widow's peak, gave them the gene for not having a widow's peak, okay? So we'll say that the dark chromosomes are, what did I say, paternal, and the light chromosomes are maternal. Now during prophase 1, they synapse, right? It's during metaphase that they line up with their homologous pair to form that, they're in that tetrad, but they line up in pairs. Okay, I always picture square dancing for some reason. They're like do-si-doing to the middle of the cell, holding hands. But it is, again, for like the 100th time, it is completely random which side of the equator they wind up on. When I grabbed these socks and lined them up like this, I just happened to put the paternal pink chromosome on this side and the maternal pink chromosome on this side. I put the paternal on this side, maternal on this side, paternal on this side, maternal on this side, right? I could have done it like this. I could have done it like this. I could have done it like this. I could have done it like this, right? So with 3 sets of pairs of chromosomes, there are 8 different combinations for how the chromosomes line up, okay? You can actually go through each one, right? Let's, here's one with all the maternal on one side and the paternal on the other side. Here's 2, right? Here's 3. Here's 4. Here's 5. And you can go through each one and see all 8 combinations. In human cells, there are 23 pairs of chromosomes. So imagine 23 different pairs of socks, right? 23 different homologous sets. With 23 pairs of chromosomes, there are over 8 million combinations, okay? The math is 2 to the n. The n is the exponent. So with 3 sets, it's 2 to the third, right? And that would be 2 times 2 times 2, which is 8. With 2 to the 23rd, which is what a human cell would be, it's over 8 million different combinations. So that is one way you get genetic diversity, because then during meiosis 1, these homologous pairs would separate. And it just so happens that you had, right, these paternal chromosomes on this side, and this one is maternal, this one's on this side. Then during meiosis 2, these would get split up, right? And this would be its own cell, this would be its own cell, this would be its own cell, and this would be its own cell. But if these chromosomes had lined up differently during meiosis 1, during metaphase 1, let's say they lined up like this, that completely changes the outcome of the cells, right? So that's meiosis 1 and then meiosis 2. Now, this is a completely different cell. These are a completely different set of genes than what you had at first, okay? So this is another way that cells become genetically diverse. The gametes, specifically the sperm and egg, this is how they become diverse. Between independent assortment and crossing over, the number of possibilities, the number of combinations is infinite. No 2 gametes produced in a human's lifetime will ever be genetically identical. When you combine the effects of crossing over and independent assortment, the possibilities are infinite, limitless, okay? So that is meiosis. Hopefully this cleared some things up. This will be our last lesson about meiosis. We're not going to have like a test or any formal quiz or anything. So if you're still confused about something, please reach out. This concept is very important to understand if you plan on going into any higher level bio course in the future. All right, hope you're all doing well. Stay safe. Reach out with any questions. Take care.